Hi, good evening one and all. So now I'm going to discuss about one of the most complicated system, that is complementary system. So this video can be used not only in the exam point of view. Yeah, hi everyone. So now I'm going to discuss about one of the most important topic in microbiology. Most of you feel it is as a complicated system. Okay, so let's talk about the details in the complementary system. So this video is going to be very useful for all the students, particularly the second year BDS students for the university exams, and followed by the first year MDS students for their basics exam. There are few departments in which this question is again a most commonly asked question, and of course it is very important in a NEET or AIIMS or any of these PG entrance point of view. So basically, the first important aspect that we have to know about this is, what is the use of this? Okay, what is the use? So regularly, our bodies contains this complementary system in an inactive form, and this complementary system will get activated whenever an antigen that is entering into the body. For example, there is an infection, so there will be an entrance of this antigen into the body. So it's going to react with antibody. So this antigen antibody reaction gives rise to the formation of antigen antibody complex. So to protect the body from the infection, this complementary system is going to come and act, and it's going to lyse the antigen that has already entered into the body. Okay, so this is what the basic idea of complement system means. Everyone has complement system. So when it's going to active means whenever an antigen that enter into the body, this system is going to get activated. Okay, so what happens? So what happens in this is very simple. And coming to the activation of this complement system, okay, there are 3 complement systems according to the textbook. The first one is called as classical. The second one is alternative, and the next one, that is the last one, is lectin pathway. So we have 3 pathways of complement system. One is classical, second one is alternative, and third one is lectin mediated pathways. So what is the advantage of classical? Why it is called as classical means because it is first identified. Okay, the classical is the one which is first identified, and it is more recently with lots of modifications. Lots of modifications are done in the classic pathway, and the classic pathway is mainly concerned with specific active immunity, whereas your alternate and lectin mediated are more primitive ones, means more old ones. No modifications were done, and most of these are dealing with non-specific innate immunities. So your classic is mainly concerned with specific active immunity, whereas your lectin and alternative are mainly concerned with a non-specific innate immunity. So this is what first basics that you have to learn about the classic, alternative, and the lectin. So the next one is, what is classic? So this presentation was made as simple as possible, first to understand the basics. Okay, then you can go for the deeper levels if it is required. Okay, so first is classic. Word classic seems to be very cool. Okay, so classic is mainly concerned with, the most commonly asked question is, classic complement system pathway is mainly concerned with which are the immunoglobulins. Okay, the answer is IgG and IgM. You can remember it as GM. Okay, my code for this is general manager is classic. So classic system is mainly concerned with IgG and IgM. Out of these two, the most important one is IgM. If you have both the options, go for IgM as a better option. IgM is more oriented to the classic, both as well as IgG and IgM are mainly concerned. If you want, you can add two important MCQs or two important points here. IgM is the largest among all the immunoglobulins, and IgG is occupies the maximum concentration in the list of immunoglobulins. Okay, maximum is IgG and size is larger in IgM. So both this IgG, IgM, that is general manager is classic, are mainly concerned with the classic complementary system pathway. So what happens in the classic complementary system pathway? As already discussed, for a complement system to get activated, there should be someone should enter into the body. That someone is called as antigen. So antigen once enters into the body, our body will produce antibodies. So antigen antibody leads to the formation of antigen antibody complex. So body is ready with the antigen antibody complex. So antigen antibody complex is going to convert C1, okay, convert C1 to activated C1. So antigen antibody complex has converted C1 to activated C1. So activated C1 is going to act to C4. Whenever the C4 is activated, C4 is going to split into C4A and C4B. Okay, C4A and C4B, in which the C4A, A for away, simply to remember, A for away, it goes away. C4A, it goes away. And the C4B is moving to the next level to get activation of C2. Again, C2 is going to split into C2A and C2B. Okay, where this C4B is going to combine with C2A. Okay, it's going to combine with C2A and it forms C4B and C2A complex. You can call it simply as a binary complex because it has two molecules in it. So this C4A is going to activate C3. Again, C3 is going to split into C3A and C3B. Hope you're clear with the classical one. Classical one is mainly concerned with IgG and IgM, mainly concerned with IgM. Whenever the antigen antibody enters, it reacts to form a complex which is going to activate C1. C1 is going to activate C4. C4 is going to split into A and B. A goes away, whereas B is going to activate C2. And C2 is going to split into C2A, C2B. Therefore, C4B and C2A combines to form a complex called as a binary complex, which is going to activate the C3. Okay, so the main purpose of the classical one is to activate C3. Okay, so the second one, we are done with the first one. So we are going with the second one. The second one is alternate. The second one is alternate pathway. So in this alternate pathway, the activation is mainly done by the either endotoxin. So endotoxin is going to help in the activation, or your IgA. Okay, so either endotoxin or IgA, because we have already discussed that classical is mainly concerned with IgM and IgG, whereas your alternate is mainly concerned with immunoglobulin IgA or the endotoxin. So this endotoxin is going to activate your C3 and the factor B. So it's going to activate C3 and factor B. So once this endotoxin IgA are going to activate these two components, as already said, they're going to split. Factor B is going to split into factor BA and factor BB. Similarly, C3 is going to split into C3A and C3B. So what's going to happen? So the molecules C3B and factor BB, they combine to form a binary molecule called as C3B and BB. So this C3B and BB leads to the breakage of C3, because the ultimate goal of any complement system, any type of complement system, lead to the breakdown of C3. We have already discussed the classical one. The end product of the classical one is the breakdown of C3. The end product of alternate is also the breakdown of C3. So the next one is the third one. What is the third one? So we have already discussed the classical, we have discussed the alternate pathway, and the third one is lectin mediated or lectin pathway. Okay, so what is this? You need to learn about one point related to the lectin pathway. Lectin is the one that's going to activate the complement system binding to mannose residues. Okay, they binds to mannose residues. These mannose residuals will present on the surface of bacteria. So whenever the bacteria enters, it's going to detect that mannose residues which are present on the surface of the bacterial cell wall, and this is going to come into action. So that's the reason why it is called as MB lectin. So it is called as MB lectin. So this MB lectin is going to bind to the bacterial cell surface. So you have your bacterial, the bacteria has the cell surface. On this cell surface, you're going to have these mannose residuals. So this MB lectin is going to bond or attach to the mannose residues which are present on the surface of bacteria. If you want, you can write down the names of these bacteria. That is the bacteria Salmonella typhi, Neisseria, Listeria, Cryptococcus, and you can add a note about the fungus also, that is Candidias. Okay, so all these, they contain these mannose residues on the surface. So whenever these going to come into action, the MBL is going to activate. So once this activation is done, this MBL is going to lead to the activation of a specific proteases. Okay, there are few specific proteases like MASP1 and 2 which are present in the body. So these are going to get activated. These are specific proteases. So this is in turn going to activate the C1. This is already discussed. The C1 is going to activate in the classical system, the first one. Similar to that, the C1 is going to get activated. Once C1 is going to get activated, similar like your classical one, the C1 is going to activate C4, which is going to release into the C4A and C4B. The C4 is going to activate the next one, that is C2. The C2 is going to activate the C2A and C2B. So overall, there will be formation of one binary complex, that is C4B2A. which leads to the breakdown of C3. The target of us is breakdown of C3. So the first classical system, the target is breakdown of C3. Alternate system, the target is breakdown of C3. And last one is the lectin pathway. Even the end product of lectin pathway is the breakdown of C3. So now we are going to learn the common complement system. Means this is common for everything. What we have learned is the difference between 1, 2 and 3. But now it is combined. Means all the classical one, the alternate one and the lectin one, the end product of all is C3, C3, C3. Now we have C3 in all this. So we're going to learn what is going to happen to C3. So we are with an end product of C3 or activation of C3 or breakdown of C3, and C3 is going to break into C3A and C3B. So we got the end product. The end product is leading to the breakdown of C3A and C3B. So C3B, A goes away. Simply to remember, you can remember A will go away. So the next one is the C3B is going to activate the breakdown of C5. So similarly, the C5 is going to break into C5A and C5B. So as already discussed, A goes away. So what is the job of C5B? C5B is going to activate. C5B is going to call all the friends. Okay. So until now, the C1 has activated C4, C4 has activated C2, C2 has activated C3, C3 has activated C5. So they have discussed all the complement components as discussed. Yeah, we are getting bored. One activating other, other activating other. So the time is running off. So C5, let's call everyone. Okay. So C5 has called everyone. Whoever are there, whoever are involved to help the body, please do come. So the C5 now has called C6, called C7, called C8 and called C9. Okay, all these people, they combine to form a beautiful complex called as, it is called as C5B9 complex. Okay, this is called as C5B9 complex, or you can simply write down it as C5B6, 7, 8, 9 complex. So this complex is given a beautiful terminology called as MAC. MAC is nothing but membrane attack complex. So what do you have? So you have your antigen. Now the complement system has given MAC. So MAC is membrane attack complex. So it's going to convert the antigen into something like this. It's going to split or it's going to break the membrane, and finally this MAC is going to help in lysis. So this is what basically the complement system. Okay, so you have classical, which is mediated by IgM, and alternate, which is mediated by IgA and endotoxins, and the last one is lectin mediated. The target of all these three complement system is origin of C3. So what C3 is going to do? C3 is going to activate C5. C5 is going to call all the friends and form a complex. The complex is called as MAC complex. MAC complex leads to the breakdown of antigen. That is the lysis of antigen. Okay, so this is what the complement system is all about. Okay, so what happens? Like we have so many proteins that we have discussed in the complement system. That is the C1, C2, C4, C5, C3, 6, 7, C8, C9 and everything. So these are called as complement proteins. Proteins which helps in the sequence of occurrence of the complement system. So there is something like there are few diseases which are caused due to the deficiency of the proteins. So it's better we split the proteins first of all. Before we talking about the deficiencies, it's better we split the proteins. We split the proteins into early proteins, mid proteins, late proteins. Early proteins are the first one. What are they? C1 activated, C2, C4. The mid protein is the goal of all the complement system is to activate the C3. And the last one is the late proteins are C5, C6, C7 and C8. Clear. So what is going to happen if there is a deficiency of early proteins? So whenever there is a deficiency of early proteins, these early proteins are nothing but C1, C2 and C4. There will be high chances of occurrence of autoimmune diseases. So there is high chances of occurrence of autoimmune diseases, and the best example is SLE. Many autoimmune diseases are mainly concerned with the activation or the deficiency of this early proteins, that is C1, C2 and C4. So next one is the mid proteins. So whenever there is a deficiency of mid protein, the mid protein is nothing but C3. So whenever there is a deficiency of mid protein, there is high chances of occurrence of acute bacterial infections or pyogenic infections. Okay, the next one is the deficiency of the late proteins. What are these late proteins? The late proteins are 5, 6, 7, 8, yes, of course, 9 also, right? So whenever there is a deficiency of occurrence of late proteins, and most commonly it may lead to the infections of gram-negative bacteria, mostly gram-negative Diplococcus, and it may cause an infection with toxoplasmosis. Okay, right? So these are the mainly concerned. Whenever you have a deficiency of any of these, for example, 5 is activated, 5 is going to call 6, 7, 8 and 9. So when you're calling the leftover people, few may be absent. Like for example, 6 may be absent, 6 may not be interested, or 7 may not be interested. Okay, 6 may not be interested, or 7 may not be interested, or 8 may not be interested, or 9 may not be interested. Even if one of these complement system is absent, for example, if there is an absence of C9 or C8, only C9 or only C8, even then there is no problem, because the rest of the people can form the complex, and this complex is going to act as MAC, and this MAC is going to break or lead to the lysis of your particular antigen that has entered. I hope you're clear. I'm just going to recap it again and again. You need to practice this again and again. Okay, so we are clear with the classical one. The classical one is basically is associated with GM, general manager, and mainly concerned with M. The goal is to activate C3. And the alternate, okay, so the alternate one starts with endotoxin and IgA. So endotoxin and IgA are going to activate the C3 and factor B, so form a binary complex which is going to lead to the breakdown of C3. And the next one pathway is lectin pathway, which is mainly concerned with MB lectin. So it's going to bind onto the surface of few bacteria, and it's going to activate MAS P1 and P2. Further, it's going to activate C1. C1 is going to activate C4. C4 is going to activate C2. C3 will lead to the breakdown of C3. So you got C3 in all the systems, that is classical, alternate and lectin. So C3 is going to break down into C3A, B. It's going to activate C5. C5 is going to call all the friends and form MAC. So MAC is going to cause the lysis. And your proteins are divided into 3 types: early, mid and late. Early, autoimmune diseases; mid, bacterial or pyogenic infections; late is gram-negative bacteria, toxoplasmosis. Even if there is a deficiency of any of these late, okay, that is the end part, like 7, C9 or C8, there is no problem. The rest of the people are going to form this MAC, and that MAC is going to break the protein, break this antigen and cause the lysis. Yes, done for now. All the best. Study well, guys. Signing off, Dr. Srikanth from